Thank you. Okay, thank you very much, Fred. So I know what you're all thinking, because I am thinking the same thing. It's the end of the day. Who the hell is this clown? Well, I am the clown that drew this spot. So don't get on me, because I'm 51 years old, and according to my body clock, it's 6:15 p.m., which means I should have my jammies on at home. So let me power through this, and then we'll all be able to get to the reception afterwards. So I really appreciate the invitation, Fred, and it's a pleasure to tell you about our work on thinking about the gut microbiome as a source of therapeutic drug targets. So what I want to start with is that this is the paradigm. And for humans, antibiotics changed everything. Not just because we could cure disease, not just because you lived when you used to die, but because this proved to humanity that we could cure disease and we could understand how we did it at the atomic level. So the structure of this antibiotic was solved at the atomic level by Dorothy Hodgkin at Oxford. The target of this drug was subsequently solved in the years later, but we now know at the atomic level how everything works. And this built the pharmaceutical industry. All the big drug companies that exist today made their money on these drugs. And as we know, they've all shifted on to other things, but this invented and proved molecular medicine as a way to do things. And our thought on this is that even though that proved the paradigm, we've missed a paradigm that's been sort of lurking there. And this paradigm didn't really become apparent to us until, just like the technology that helped show that antibiotics worked, we didn't have the technology to really see the microbiota that existed on and in us. And then we now have that. And so my overarching thing is that within the 4.8 million proteins that are expressed or have the potential to be expressed in the gut microbiome, there exist druggable targets. And those are just as interesting and just as tractable as the 23,000 genes that are in our genome. And so what we've tried to do is come up with this set of rules for, if you're going to do this, let's do this right. We want to have drugs that are potent. We want to have them that are selective. We only want to hit certain enzyme targets in the bugs and no enzyme targets in us. And we want to have them be non-lethal. And that's really important because we don't want to kill the bugs, as we've heard many great examples of. And so this is what we're up to. And so just another slide to sort of set the stage and just to show you that, of course, there are all these diseases and phenotypes that are associated with gut microbiota. And it's not clear in some cases whether or not there's a cause and effect. I'm a biochemist. We really like simple things with reversible arrows, so you're going to see a lot of those. And where we come in is at the bottom of this list, which is in perturbing this system with very toxic drugs and seeing what role the microbiota play in the outcomes with that and how we can control that. Okay, so the drug I have to introduce you to is irinotecan. This is a widely used chemotherapeutic for pancreas cancer and for colorectal cancer. It's the IRI in the FOLFIRI or FOLFIRINOX regimen. It's given to patients as a prodrug. It's infused in looking like this. It gets activated in us to its active form, which goes off and poisons topoisomerase 1, which is overexpressed and overactive in rapidly dividing cells. Your body doesn't want to keep this around, so it metabolizes it by putting a sugar on it. That then marks it to be sent to the gut. This is a normal metabolic pathway. In the gut, there exist microbes that, just like my kids, love sugar. And they chew this sugar off. They scavenge it off as a source of carbon. And that reactivates the drug in the lumen of the gut, and that produces the dose-limiting side effect of irinotecan, which is acute delayed diarrhea. And so the single step we made in this project was to say, well, and in fact, we'd walked every one of these arrows through this, because I've been interested in irinotecan for 25 years. We finally said, why don't we just follow it into the microbial compartment and look for druggable targets in there. And it turns out there's a single enzyme class that I call GUS. That stands for beta-glucuronidase. You have a GUS. It's an enzyme. It just cleaves sugar off. The bacteria have GUSes as well. Turns out they're different. Your GUS is essential. The bacterial GUSes are non-essential. They're scavenging enzymes. And so our thought was, why don't we try to drug the non-essential GUSs that are scavenging the sugar off of this anti-cancer drug. And so quite a few years ago now, we did this and we proved sort of the first concept of this. We did high-throughput screening with a GUS from bacteria. We found potent inhibitors. One of them is shown here. We then set up a mouse model of irinotecan-induced gut toxicity, and we showed that we can get delayed toxicity in the mice over about 10 or 11 days. All the mice get diarrhea. And if you treat the mice with this inhibitor orally, a couple times a day at actually a relatively small dose, you can prevent that gut toxicity. And it's really shown here in the histology quite well. You can find even a 5-year-old pattern recognition machinery would be able to find the mouse colon that was treated with irinotecan. There's no glandular structure left. It's completely destroyed. All those blue cells are inflammatory cells. And if you give our inhibitor on top of that, you partially protect, not completely, but as a proof of concept, we partially protect the damage to the gut. And it turns out that about 30% of human patients who receive irinotecan have grade 3-4 chemotherapy-induced diarrhea, which is terrible. I'm not going to take you through the grades, but you don't even want grade 1. Grade 4 is life-threatening, so this is bad. So this is our paradigm now, is that within the gut microbiota, within the trillions of cells sitting in there, there are enzymes that we can come in and inhibit. And during a patient's chemotherapy, we would use potent, selective, and non-lethal compounds to do this. And it turns out the compounds we got, you got to be lucky. I drew this slot, last one of the day. I've got to be extra energetic to keep you awake. We found compounds that were completely selective for the bacterial enzymes. And so we got selectivity. We don't inhibit the human ortholog. Okay, so in doing this, what we ended up addressing or modulating was a natural enterohepatic cycle, where you have lots of different compounds in you that are marked with this particular sugar and sent to the gut for excretion. The enzymes in the gut, the gut bacteria evolved enzymes to scavenge these things off, and so there's this natural cycling pathway. Turns out there's hundreds of different compounds that are sent to the gut with this sugar attached. And so we're curious about the many different ways we can try to think about using this technology. So it turns out irinotecan is not the only drug that causes this problem. Non-steroidal anti-inflammatory drugs do as well. And so here's an example of one, diclofenac. And so that's just an anti-inflammatory drug, and it's metabolized in the same way with this sugar attached in the liver, marks it for excretion into the gut. And in the gut, these GUS enzymes scavenge off the green sugar, which are like the green M&Ms, I guess. It's like my kids love those. So creepy that I said that. And it turns out that for NSAIDs, that gut toxicity is not diarrhea. It's not in the large intestine. It's in the small intestine, and it comes in the form of ulcers. And so we set up a collaboration to try and address this with a great, now retired pharmacologist at Connecticut named Urs Bolsterli. And so if you give mice diclofenac in the center image, you can see you'll get these longitudinal small intestinal ulcers. And if you treat with our inhibitor on top of that, you significantly reduce that, that damage in a different part of the GI tract. Well, of course, NSAIDs are the most widely used drugs in the world, 30 billion annual doses just in the US. And it turns out that with video capsule endoscopy, you can now see these damages. The small intestine is the hardest place to get to. You can't really get a tube through either way to get there. So you swallow a camera and you can take pictures. And 40% of long-term NSAID users have small intestinal ulcers. So what I've told you is that the microbial GUS enzymes are the first drug target that was discovered in the microbiome. GUS inhibition prevents toxicity from both irinotecan and from NSAIDs, and our inhibitors have those characteristics. And so what I want to do now, I'm a protein biochemist, I'm a structural biologist by training, and so we're going to do some crystal structures, we're going to look at some active sites, and what I'm going to tell you is that there's an amazing amount of diversity in this drug target within your gut. So if you think about the kinases and all the different kinases in humans and all the different splice variants, you could think that's complicated, but grab a hold of something, because we're going to go right into the gut microbiome. I'm going to show you how complicated that is. So if you're going to drug a target, you have to know things about it. So we ask the question, how many different types of GUSs are there in the gut microbiota? And what we tried to do then was set up an effort to define the Gus ohm. If you're cool, you got to have an ohm. You got to roll with some ohmies. And so this is our ohm that we defined. And the thing we wanted to find was any enzyme that we could identify using structural biology rubrics that could catalyze this simple reaction, which is the removal of a glucuronic acid from this ether linkage. And so what we did was we took a bunch of the crystal structures that we'd solved of bacterial Gus enzymes, and we defined what was necessary at the active site for it to bind to its substrate and cleave this bond. And it turns out it's a very precise set of amino acid side chains. And you could develop a really clear rule that any protein that looked like this and had this at the active site was a Gus. And then we went to the HMP and we just mined through their 4.8 million genes that's in their stool sample database. And we found all the Gus's. And so it turns out the HMP is organized in a couple different ways. There we go. There's sort of, when you took the stool samples, there's sort of everything. And then they said everything's too much, and so they clustered it by 95% sequence identity. And they said, well, here's the unique stuff. So I'm going to start with the unique stuff. That's the 4.8 million genes. Take all those genes and you put it through this filter that we developed, and it turns out there's 279 unique, non-redundant Gus enzymes. That's our family of drug targets. It's our first definition of the family of our drug targets. And so here's the structural biology. So here's where we got lucky, which is when we did our high-throughput screen to find our first inhibitors, we found this inhibitor was one of the first, and this is how it binds. And it binds at the active site of the bacterial enzyme using a big loop that's in red. And that loop is missing from our enzyme, from the human enzyme. And so that's where we get our selectivity. It turns out the loop is where these enzymes sample their functional diversity. So we're going to look at this bacterial loop, and we'll let this rotate here. And so this is how the loop sits near the active site. The catalytic residues are in pink. Some really important fingerprint residues, blah, 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 are in a different color. And so everything matters in terms of how you decorate this active site. So here we go. So it turns out there's 6 different categories of Gus enzymes in the gut microbiome. And so when I talk to Curtis Huttenhauer or Rob Knight and I say, oh, this is what we're doing, you're sequencing all these things, but we're going in and we're applying these rules. And they're like, whoa, a Gus is a Gus. Like when we define it as a Gus, dude, we're out. We've done our job. I'm like, no. It turns out that all the information you need to figure out what it does is still there. And this is all not the same gene. These are different genes. These are different proteins. They do different things. And so to sort of play that out, I'll tell you that the ones that have this big loop that I showed you first, which we call loop 1, are the ones that process small drugs and lead to adverse drug reaction events. And those are the ones we can inhibit. That's one group out of 6. The other 5 have very variable active site architectures. They do different things. They don't work that well on small molecule drugs, and they seem to process polysaccharides. We don't inhibit those. And so we're getting some granularity about what's happening within the gut microbiome. And so we call this analysis metagenome analysis by protein structure, or MAPS. And the first paper we published on this was an atlas of what the Gus's looked like in the gut microbiome. And so we're interested in carrying this forward and looking at any number of other enzymes that are expressed, or proteins or factors that are present in the gut microbiome that we can now annotate and really get down to the level of precision health using this sort of information. Who might get poor outcomes with irinotecan? There's no human gene that defines why 30% of patients have poor outcomes and 70% of patients seem to do fine. We would argue it's their gut microbiota composition. And in fact, if you go away from the simple part of the stool sample catalog and you go back to the original part, this original part came from 139 healthy, probably med students at WashU and Harvard or wherever they recruited these people. And you can break out their gut metagenome by person. And then we can, for each individual, 1 to 139, we can find all their Gus's. And we can figure out what each person's Gusome looks like. And if you do it that way, with all the redundancies present, you have a little over 3,000 different Gus variants. But the cool thing is you can start to ask the question, what can this tell us about precision health? And so, first of all, everybody who was sampled in this data set had a GUS. You have between 4 and 40 of them. Average is about 23. So it's an enzyme that's present in your gut. It's doing things. If you stack this up, though, and I have my 6 different categories in a different color, you can see that everybody's got sort of a little bit of a different pattern of how many GUSs they have. And interestingly, about 60% of patients, or 60% of these donors, have one of these loop 1 GUSs. And that actually corresponds with, if you look at the total amount of gut toxicity, there's about 30% of patients that completely tolerate irinotecan with no even grade 1 diarrhea. But about 70% of them show some gut toxicity. And I would argue the people on the far right, the one that has about 30% of their GUSs are loop 1, would be the ones that might have poor outcomes. So we're in the midst of testing this at UNC hospitals. So the real question then, in terms of something to think about for our future work, is what does this really tell us about health, and what can we do with this fingerprint of information that people are flushing away on a regular basis? Can we get information about this in terms of clinical outcomes? So there's a handful of different types of GUSs in the gut. There's a few hundred of them, and they're specialized for different types of substrates. Okay, so this is my last data slide, so be cool. We almost have our jammies on, or we're about to drink, or whatever we're going to do. And so the reason I started this was not because I didn't want people to have diarrhea. I certainly don't. What I want is good outcomes. It's hard to find drugs that kill cells, that kill rapidly dividing cells. Irinotecan's a great one. Can we use what we're doing to make its efficacy and safety better? So we used a breast cancer gem, and what we did was dosed up this gem with irinotecan, and we asked, do we get less diarrhea? And in the green versus pink in the center, you can see that all the animals get diarrhea within about 14 days with just irinotecan. If you add one of our inhibitors on top, we can protect them from getting diarrhea. The Kaplan-Meier curve in the upper right looks good too. We can extend the life of these animals. But the thing that got us the most excited is that in terms of tumor regression, we can significantly reduce the size of the tumors. And the reason for this is that the inhibitor doesn't do anything to the tumor. It just keeps the gut intact and healthier so that the mouse can see more doses of irinotecan. And in fact, when the animals are dying here, we're having to euthanize them for cachexia, which is just toxicity that's off gut toxicity. It's just muscle wasting. And so our current goal is to redo this study and try to get a Kaplan-Meier curve that stays straight all the way around the corner. And really sort of get the tumors to disappear while the animal stays alive. Okay, so here's our summary. Is we can do this for cancer treatment and other adverse drug reactions and maybe even other disease states. So a lot of needle in the haystack today, and so here's another one. So we feel like we found the first needle in a haystack that's going to contain other druggable targets. And here's my disclosure slide. So since 2012, we have licensed this technology to our startup company, which is now taking this into a preclinical stance and then heading toward an IND to bring this to patients. And here's a picture of the building I get to work in. And the people that are the real heroes here are Brett Wallace and Audra Bott, who did most of this work, as well as Rebecca Pollitt, who's sort of in yellow down near the bottom. And thank you very much for your attention.